Well, good morning, good afternoon, and good evening, ladies and gentlemen. My name is Brad Sayles, and I am the Chief Operating Officer here with Semex, and we're very pleased to be bringing you this webinar today. A few housekeeping items before we start. First of all, within the mechanics of the webinar, you'll see a spot to enter questions. We do have all of the microphones muted for our webcast today, just to maintain some order, but we welcome any questions to come through that feature that should be present on your screen. And if you can't see it, you probably need to push one of those little sideways arrows up at the top, and that window should open up for you. So we have quite an exciting presentation today. I think Semex has been very fortunate over the years to have an outstanding relationship with the University of Guelph. Semex has been a big supporter of research over the years, and most importantly, research that will result in things that can have real practical applications on the farms of our customers globally. And one of our greatest collaborators has been Dr. Bonnie Mallard. And Bonnie, I'll get you to switch to the next slide, and we'll take a look at some of the questions we're going to run through today. But I'd like to introduce Dr. Mallard, or I guess Bonnie, as I know her. She has been a valued member of the University of Guelph faculty for some time. She's been employed in the Immunogenetics department and has conducted a lot of research and has really developed among her students a lot of research leaders right around the globe in our agriculture industry. She's received a number of faculty awards, and we've been very pleased to work with her on the development of her high immune response technology, and which eventually became Immunity Plus. And we're so excited of the progress that that technology has brought to our client herds. We're now seeing second, third, fourth generation animals being born on dairy farms globally, and there's no question there's a tremendous economic benefit to the technology. So we have 4 or 5 questions that we had asked Bonnie to go through today. And she's prepared a PowerPoint presentation that will take us through the discussion. And then kind of when she finishes up with each question, we'll pause for a few minutes, and Peter Van Beek from Semex is here to help us coordinate some of the questions as they come in. So Dr. Mallard, Bonnie, it's great to have you. We're so thrilled that you work with Semex, and we're thrilled to have you on board today.
Yes, good morning, afternoon, and evening, everyone, depending on what time zone you're in. And thanks, Brad, for that kind introduction. And it's really a pleasure for me to talk about the high immune response technology. And we'll just start with a really brief overview of what it is, what the elevator speech is, so to speak. So this is a patented test, and it provides a means to identify and breed animals. And these animals possess 2 things: inherently superior immunity or an immune system, and therefore enhanced disease resistance. And so this results in healthier herds and happier customers. So that's kind of it in a nutshell. And as Brad said, we've been partnering together with Semex Alliance for quite a while. And they actually have the exclusive license to use the HIR technology, and that's what we call it at the University of Guelph, and market it under their trade name, Immunity Plus. So I think the next piece is just to give you an overview. And let's just start with the 2 big benefactors of this, and that's the livestock producers. And they benefit from having both a management and a breeding tool to naturally enhance the health of their herds. And I say naturally, and that's really was always very important to me. I don't think consumers are ready really for GMOs. That's proven out over the years, and I wanted to stay well away from that. So I really capitalize on the animal's own genetic potential. The other benefit is reduced lost income due to illness and reduced veterinary treatment costs. And then the last point there is improved decision making around breeding and culling. The consumer is the other person that benefits. And again, there's consumer expectation around natural, improved food quality. And so this technology fits that bill. The other thing that's really important to everyone, consumers and producers alike, is this reduced use of antibiotics in food production. And so if you have healthier animals, you just don't have to use as much antibiotics, and that's kind of the beginning and the end of that one. And again, we wanted a sustainable, healthy and sustainable technology to be able to give enhanced food quality all the way through the food chain, right from the farm to the fork. And then the last point, and not the least, is really the animal well-being, because healthier animals are happier animals, and really consumers now want to know where the food comes from. They actually want to be able to trace it all the way back to the farm of origin. And so when that happens, we want to make sure that these animals are healthy all the way through.
Very good, Bonnie. One of the questions that we often get is about the transmission of this high immune response from one generation to the next. And I think it's important if you just walk through the transmission, because high immune response is not a single gene trait like something like coat color or pole, that there are a number of genes, throughout an animal that control immune response, much like milk production. There's not one gene that, controls milk production. So we often talk about, breeding for healthier animals is like breeding for milk production. If you focus on it in your breeding program, you select for it, over time you will make improvements. So maybe just a couple comments on transmission from one generation to the next.
Yeah, I think that's a key point. And so it is a lot like milk production. It's called a complex trait. And you don't just have a few genes, but you have many, many genes controlling it. And then heritability becomes important. And these next couple of slides kind of walks through the science behind that, the depths and scope of the science, and really talks about the genes. So maybe I'll kind of start there, Brad, if that's okay.
Yeah, I'll just ask Peter. Peter, do you have any questions that have come in from our audience today?
Yes, somebody was asking, so is it only technology that works for specific diseases like mastitis, or how does that work out?
Yeah, so again, we chose a technology that would not be specific to one single disease. And so mastitis is definitely in the group. And again, I think as I walk through the depths and scope of the science behind it, you're really going to see how this works, and you'll get the answer very specifically to that question.
Okay,
Great.
thanks. Yeah, that was the one we had so far.
Thanks, Peter. Okay, Bonnie, we'll move on to question 2.
Okay, so really this is an immunogenetic approach to improve immunity to dairy cows. We do work in other species, but the focus for today will be talking about the dairy cattle. And we really wanted to ask that question. Could we use a combination of knowledge in immunology and genetics to improve cattle health? And so when we started to think about this, we had a couple of things in mind. And this goes back to that question that was just asked here a minute ago. So we purposefully decided to select for broad-based disease resistance rather than on resistance to a single disease. I knew that that was entirely possible to select for resistance to a particular disease, but it came with a cost. And that cost was susceptibility to other diseases. And so I wanted to stay away from that approach. I was after broad-based disease resistance. And so that meant I really had to focus on the immune system. And the other thing that we did differently than other people had done in the past was we decided we wouldn't even just select based on a phenotype, but it had to be an estimated breeding value of that phenotype so that we were sure we were capturing the genetic information and minimizing the environmental component. So that was something that was unique to this technology and is still quite unique today. And then the third element that was unique and is still unique is that we used a multi-trait selection index. So we put a variety of indicators of immune response into the selection index so that we could improve a number of elements of the immune system. And this slide, I think, helps to illustrate that. So first, we focus on the immune system because that's the only system that the body has for host defense. And if you see the triangle here at the bottom, this kind of tells you all the elements that makes up an individual's immune response phenotype. And that doesn't matter if you're a dairy cow or a human being, it's the same elements. And so if you look in the triangle here, there's two arms, and the first arm is antibody-mediated immune response. So that's antibody, and we're hearing a lot about the value of antibody in regard to COVID-19, for example. But there's another arm over here. This is cell-mediated immune response. That's things like T lymphocytes. We're hearing a little bit about that. And then this component here is innate immunity. Now, these two arms of the immune system, they have immunological memory. And that's why when you see a bacteria or a virus the second time, you're going to react much faster. And that's why we really wanted to focus on these two arms. Innate immunity is important, but it doesn't have memory. So the thing to remember here is that antibody, it is important in dealing with extracellular pathogens, pathogens that are outside the cell. And so that would include things like mastitis, you know, the E. coli, the Staph aureus components that cause mastitis, or some of the pathogens that cause scours, for example, in calves. But we also wanted to talk and think about this cell-mediated immune system. And that is what controls intracellular pathogens, particularly things like intracellular bacteria, that would be Johne's and tuberculosis, and all viruses. In order for a virus to survive, it needs to get inside the cell and replicate its DNA. So you have to have these two arms, AIMer and CIMer, I call them. These are the twins of the adaptive immune system. And they're controlled by the genome that you see down here at the bottom, and the epigenome as well is important. So we needed to focus on all these elements and measure this big P at the top, which is the phenotype. But we also wanted to keep in mind passive immunity. So as you know, calves, when they're born, their immune system is immature. And so they have to rely on high quality colostrum from their dam to have that so-called passive protection until their own immune system develops. So these are all the components that the high immune response technology wants to capture. Now, someone asked about, well, I guess it was Brad who asked about the number of genes. And so here you see that. Approximately 5,500 genes are involved in host defense. And all mammals, again, including humans, only have about 23,000 genes. So if you look at the circle here, you can see the proportion is about 24% of the genome goes towards immunity or keeping an individual healthy. And at first you might think, wow, that's a lot of use of genetic resources. And it is. But the important thing to keep in mind is that's how you survive. That's how a person, an individual survives in order to be able to pass its genetics on to the next generation. So that's why we focused on the immune system. But we have a lot to learn about all the genes and how they interact. And we know a lot about that now. So we were able to develop a test, a phenotypic test. It's a 15-day test. You do it on farm, 3 farm visits, and at the end of the day, we get a very accurate measure of antibody, as you see over here, and cell-mediated immunity, as you see here. So you can see the circle there, that's a positive reaction, and where the calipers are here, that's the negative or control in the tail fold. This is indicative that an animal has allowed the cells of the immune system to infiltrate that site. So it's a little bit like a tuberculin test. And it says, yes, this animal can respond and make cell-mediated immunity. Here, this is a picture of one of my students, and that blood sample will go back to the lab, and then we will look at antibody. And we have our 2 indicators now of immunity. What about heritability? This is key. So when we were devising this particular technology, we wanted to only have traits in our index that were quite heritable. And so you can imagine we did have to screen a lot of traits, a lot of test antigens. We had to take a lot of time to really get the protocol that we use today. But you can see the heritability of the AIMer and CIMer traits that I just showed you is in around that 30%. This is moderately high. And if you compare that to production, the heritability of production traits, for example, or conformation traits, you can see that here, it's right in the same ballpark. So that's why Brad said he often thinks about this in terms of the same way as you try to improve milk production, you can improve immune response. And that's exactly correct. Conformation traits, you can see here, they're also fairly easy to improve because of their high heritability. But if we look at the bottom here, you can see there are a number of traits that are of lower heritability: longevity, calving ease, daughter fertility, and then this one here is one I want to point out to you, because this is disease incidence. So if you try to improve disease resistance by looking at the incidence of disease rather than the immune system mechanics, the heritability goes way down, because this is based on whatever records might be available and how accurate those records are. Whereas if you test the function of the immune system using a patented test system, which is highly controlled, the heritability is much higher because you're right next to the trait that's controlling host defense. And so that's why we can make really good genetic progress in terms of immunity. So with this tool in mind, now we can classify animals. So immune responsiveness is a complex trait controlled by many genes, as I told you. And these traits follow a normal distribution. That's like this mountain or hill that you see here. That's the normal distribution. And every dot here would represent an individual animal. And you can see the vast majority of animals here are average. And that's just to be expected. But over here on this side, it's on my left, you can see these low responders, and they are kind of the yellow balls here. And what we found out over the years is, unfortunately, these animals that have low AIMer and CIMer, or low immune response, and this is based on their estimated breeding values, remember, these are the ones that get sick more often. But conversely, if you move to the right, you see these dark blue balls, and these are the animals that are the high immune responders. And these are the healthier ones, as it turns out. And so once we had established this, now we can look to see, are there benefits of being a high immune responder, and what specifically are these? And so over the years, we've done a lot of studies, and this picture here shows you some of the significance of being a high immune responder. And you can also see these pathogens. That's what these little guys are here. These are microbes, viruses, bacteria, parasites, and you can see they're all different. And we designed that picture that way so that you'd remember that this is not against one particular disease, but the immune system's job is to protect you and me and our animals from a wide range of pathogenic microorganisms. And so that's what we're capitalizing on. And as we've examined this over many years, we see that in fact, high immune responders have decreased occurrence of various diseases, mastitis, pneumonia, even some metabolic diseases, ketosis, and also retained placenta. They're also less likely to be seropositive for Johne's disease. They have better hoof health, particularly digital dermatitis. They have improved response to commercial vaccines, and they have improved milk and colostrum quality. These in red are what I'll spend some time talking to you a little bit more about today. But we've also seen that they have unique genomic profiles. We will talk about the genomics test. They have altered microRNA profiles in the colostrum and milk. This is epigenetics. We won't get to that too much today. Beneficial associations with herd life and some reproductive traits. We won't talk much about that, except I want to say that these are also fitness traits. And so sometimes there's genes in common between fitness traits and immune response, because that's also a fitness trait, whether you survive or not, and increased heifer growth rate. So there's a number of things here. I'll show you a couple of graphs just to give you a sense of this. So this is data from over 100 herds that we've looked at, and these would be herds where we've done research. And so that means that either the veterinarian or trained lab technologists are collecting this data. And so this is what you would see. On the y-axis is the percent disease occurrence, and on the bottom is the category. So whether the individual is a high immune responder, and we always show the high immune responders in green usually because it's a green technology. Here's average and here's low. So you can see right away at a glance that the high immune responders have about half the disease occurrence of low. And this is pretty much what we have seen consistently over and over again. I've put some references down there at the bottom for you to look at if you want to follow up a little bit more detail on any of this. We've published a lot of papers over the years, probably over 100 listed on PubMed, and you can always go in there and look that up. Here, economic benefit. So we worked with an independent agriculture company. It's called AgriStudies Inc. You can see that on the bottom, out of Guelph, Ontario. And they helped us try to estimate the cost savings of a high immune responder cow versus an average responder, for example. And so we took into account the various diseases here that a dairy cow would commonly come up against, and plus some of the benefits of vaccine and high quality colostrum. And a very conservative estimate here is $247.91. Let's just call it $250 and round it out per lactation. And so if you do the cost benefit over time, and you just say the cost of an HIR test might vary from $20 to $80 depending on what test you're using, the phenotypic test or genomics test, and then you look at the benefit, which is here. But that's the benefit you get in year one, which is a 3-to-1. But if that cow stays for year 2, now you're up to close to $500, and year 3, it's now up to close to $1,000 savings per cow. So I think that's pretty respectable, and we would definitely say that's probably the minimal benefit that you would get. We tried to be very conservative. We didn't want to promise more than what you might achieve. Well, Brad, should I take a little bit of time and talk about the genomics test now and how we kind of got there?
Yeah, I think that'd be a great idea. Obviously, having a genomic test to add in with our Elevate genomic testing program that's now pretty much available globally, I think adds a significant amount of value and really gives an opportunity for our clients to use those genomic tests not only to decide on genetic strategies, but also sometimes some herd management strategies as far as which animals will be bred to what and kept and sold and different things like that. So I think it'd be good if you could take a minute for that.
Okay, it's a great idea. So I'll tell you how we got to this. We were using the phenotypic test, and I showed you that's a 15-day test, and it requires 3 farm visits. Well, that's a bit cumbersome to do, and we realized that if we could convert that phenotypic test to a genomics test, it would have much broader application. We realized then that people around the world could test their females relatively easily, rather than having to have trained technical people like my staff come on to their farm. And so we asked ourselves the question, could we find genomic markers that lined up and correlated with this high immune response immunity plus phenotype? No one had done that, and so our group was actually the first in the world to do a GWAS, genome-wide association study on immune response. And that was proof of concept because no one had done it. And a PhD student of mine, you can see down here, Kathleen Thompson Crispy, she published this in 2014 in a very prestigious journal known as BMC Genomics. And so that was the beginning of the genomics journey. And then we started to work with Semex to see if we could refine that and get it into a marketable test. And so what you see here is data on about 10,000 cows that were genomically tested. And this is the diseases that were recorded in those herds on those 10,000 animals and the frequency of those diseases. So you can just read across the top some of the diseases that were recorded: abortion, then we had a miscellaneous category, ketosis, laminitis, mastitis, metritis, and so on. But probably maybe the bottom line, the easiest way to look at this is the difference if you're an immunity plus versus other. And that's these values right along this shaded blue line. But the bottom line is when you add this all up and you look kind of overall at the averages, the immunity plus females have 30% less disease than their herd mates. And so that would be either the average or low immune responders. And so that is very encouraging news to us. It's really showing us that the genomic test aligns very nicely with the phenotype and is much, much easier. So then all that a producer really needs to do is send some source of DNA, and that might be hair follicles, or it might be, a skin biopsy. Makes it very, very simple. So in June 2018, not so long ago, Semex again had an agreement with the University of Guelph about using this technology and launched their actual Immunity Plus as part of their Elevate platform. And I'm sure you've heard a little bit about that previously. Okay, I think that's kind of it for genomics.
Yeah, so I think before, and Peter, you can get your questions ready, but interesting, Bonnie, that, the 3 key areas of high immune response, obviously the obvious one is resistance to disease. And, animals don't have to be, wait to be milk cows before that can be a benefit. Obviously, with calf health, transition for calves from, being when they're through their weaning period and as they grow, that's obviously a big one. Colostrum, quality of colostrum that high immune responding animals produce is far superior, which is another key element. Do you want to touch a little bit on vaccines? I'm not sure we went through that, but it seems that high immune responding animals also take better advantage of the investment that our clients are making in vaccine protocols by utilizing that vaccine to build as strong a defense as possible against whatever the vaccine is for. You just comment on that for a bit?
Yeah, for sure. And later on, we'll show you a graph of the actual difference in response to vaccination. But I saw that really early on when I did some of the initial studies in swine, actually. And we chose to work initially in swine because, you know, they have large litters and short generation intervals. So it was super convenient. But it was right there at the outset that I noticed that the high immune responders were responding a lot better to commercial vaccines, and they had a lot less non-responders, because you always have some non-responders to vaccines. And so I already kind of had that in the back of my mind when I started working with cattle, and we've done some studies again, and sure enough, the high immune responders, because they have the best and most robust immune system, when you vaccinate them, it just makes logical sense that they're the ones that respond best to the vaccines as well. So you get some synergy between some various preventative methods. My lab has always focused on prevention, and that's whether that might be vaccination or it might be genetics. And when you see synergism between the management, for example, the vaccination, and the genetics, that's really encouraging. And you'll see a graphic of that in just a few minutes.
Great. Peter, how from your side, do we have some questions that have come in from our attendees?
Yeah, quite a bit. So let's start with this one. So did you you mentioned, Bonnie, that calves grow better when they're better immune responders. So somebody was asking if you have some research about it, if you found differences between growth when calves are weaned at different ages as well.
Yeah, so again, it kind of went back, that story went back to the early days when I was working with pigs. And so for pigs, a really, really important feature is growth, days to market weight. And it was astounding, actually, to see that the high immune responder pigs, they reached market weight about 10 to 12 days before their herd mates. And so again, I started to kind of wonder a little bit about this, but it was one of my students, my former students, and he decided just to look at heifer growth, and so Holstein heifers, and he did see that those Holstein heifers that were the high responders grew faster, and we've often kind of thrown around the idea of whether that's why sometimes you get improved reproductive performance too, because they're bigger and they mature a bit earlier. So that's okay. Kind of the story on that one.
Okay, and looking at age, there's somebody with a question completely more towards the end of the life of the cow. So somebody is asking, as a cow gets older, does she lose her immune system? So
yeah, that's a great question. So there is something called immunosenescence, and that's in all animals and humans too. So as you get elderly, the immune system doesn't work as well, and that's probably why the elderly are more prone to flu or things like the COVID virus. But usually you also have to have some kind of a comorbidity along with that. And that, in terms of the animal kingdom and dairy cows, those cows have to be really old before you see immunosenescence. And they also usually have to have some other underlying problems. So generally, it's not really an issue in cattle the same way it is in humans, because we do tend to call them before they get that old and decrepit.
Yeah, exactly.
Yeah.
Okay. And I've got a question here. So somebody was asking more about the testing process when you're sampling an animal for being a high immune responder or not. So do you need a big comparison group to make difference between bulls if you want to know which ones are the better responders, or does it also matter what season the bull is tested with a physical test?
Right. So we'll start with the season effect because that's super easy. Because it's an estimated breeding value, we get rid of all of that environmental noise. So it doesn't matter what herd they're in, it doesn't matter what season they're tested in, it doesn't matter what the age is, because it's the genes, it's the genetic component that we're able to grab onto. And of course, the bigger the comparison group, the better and the more accurate. And that's why we spent a lot of time getting a very big base of information, and we definitely have that now. And when you have that, because the accuracy of your heritabilities, you can see that statistically, and so we're very, very confident. But sure, you don't want to just test a few animals. You want to test a lot of animals, and you want to be able to look and calculate accurately those breeding values. Hopefully that answers that question.
Yeah, exactly. Yeah, and somebody was asking a question about what the reliability is of the genomic test for immunity.
Yeah, I'd have to go back and just look right at the numbers, but I can tell you that it's very reliable now. You'll see later on, I think we may show you some data on that, and you'll get to kind of see the number of animals that we're looking at. And you'd have to just go back to, you'll see as we go on, there's some publications. There's one I'm going to show you or refer to later on by Steve Larmer and myself that we published in Cattle Practice in 2017. That would have all of those numbers in there accurately. I don't want to throw out a number without checking.
Okay, that makes sense. Yeah, so somebody is also asking what we do for our bulls, Brad. Maybe that's a question that you better answer. So if you do only the genomic test on our bulls that we are marketing as Immunity Plus, or also still the phenotypic test as well?
Yeah, for the Elevate program in females, obviously we just deal with the genomic the test that's been developed. But for the bulls, we continue to do the phenotypic test as well. And then we combine that with genetic and genomic information available to try to give us as much accuracy as we can get. And we've been very, very pleased with it. Obviously, the genomic information on the males allows us to do selection towards healthier cows and high immune responding animals. So that has certainly helped our selection process. But for the bulls, they must be both phenotypically and genotypically tested as well.
And I can just jump in there and also add that with all of the research that we do here at the University of Guelph, we also now do both tests, both the phenotypic and the genomic test. Again, that kind of gives you the maximum accuracy, as Brad was mentioning.
All right.
Bonnie?
Thank you. Yeah.
Okay,
Let's
thanks,
continue.
Peter. Bonnie, I know you didn't do all this by yourself, and I know it took a long time. So maybe just comment on the size of your team, and I know there was some interchangeable parts as time went by, but I know you've got a great team there at the university.
I do, and I'm super proud of them. Like you say, they change over time because we're always training students, and we've got technicians, and this is kind of just a picture I took of the team probably 2 years ago, and so you can kind of see the size of it. And always we have postdocs, research associates, and then we have PhD students and master's students and undergraduate students. So it's a real education process, but we always have experts in immunology, genetics, and immunogenetics as part of the team. We always need those three components if we're going to keep at the sort of the cutting edge of our game. I'm also a member of the Center for Genetic Improvement of Livestock at the University of Guelph, CGIL. It's in the Department of Animal Biosci, and I'm in the Vet College, and so I'm a member of both of those groups. And so that's the other reason we're able to marry these disciplines very, very effectively, because we're right across the road, and then Semex, our partner, is just down the street. There's not very many places in the world, actually, where you've got all those team members you've got a good business partner. You have also a large research station, which we have, which is about 20 kilometers from the university where we can do all of this. So yeah, and then you need funding. So again, I've been really fortunate to have funding, and you can kind of see all the logos here, Genome Canada, MyTax, and Semex, and all along NSERC, all along the bottom. So yeah, I definitely did not do this by myself. That would be an understatement. It wouldn't have happened.
Okay, that's great.
Oh,
Yeah, go ahead, Bonnie.
I just wanted to say a little bit about the awards too that we've won, because this goes to the credit of having a really effective team and a great partner. And so I'd really be amiss if I didn't say, you know, we have gotten 7 awards as a team, as a group since 2013. Pretty amazing. And that was another group effort. You can see some of those highlighted here, but I do want to just mention one that I'm particularly proud of, and that's this one here. This was the Governor General, David Johnson, the Honorable David Johnson in 2017, and we won the Governor General's Award for Innovation for this technology. And the thing that's special about that is that this was the first time that this innovation award had been given in the agri-food space. And so that really spoke volumes to me about the quality of this and the importance of agriculture to our country and to the world, really. So yeah, I just wanted to mention that.
Great, thanks, Bonnie. And I think we'll keep moving along here. Obviously, if you move to the next slide, one of the biggest benefits of Immunity Plus is, less animals that are, undergoing some sort of a treatment on the dairy farm today, and probably means more milk in the bulk tank and so on. So Bonnie, if you just want to talk a little bit about immunity response and antibiotic use.
Yeah, I mean, I think that's one of the biggest advantages, because we're just not able to use antibiotics the way we used to. We have to be very cautious about what antibiotics we use and when we use them. So anything you can do to keep animals healthier is going to improve that. And so I've just got a couple of slides to show you here. The first one I chose was on mastitis, because mastitis traditionally has been a place where we've used a fair amount of antibiotics in the dairy industry, and we're not allowed to do that now as freely as we had in the past. So this was a study, a research study that I was heading up, and it involved 58 herds across Canada. And we had veterinarians going in and very accurately scoring mastitis, the pathogens involved, and the severity of the disease. And you can see here the category, low, and this is antibody I'm showing you here. But if we do the calculation, this is the incident rate. And if I just do the calculation in reduction of mastitis, you can see here the overall reduction, if you're a high immune responder versus others, was 35%. And so there you have a really big savings, not only in terms of dollars, but in terms of treatment costs. Now, the other thing I want to point out before I leave this particular slide is the severity, because this was probably one of the first times we had the opportunity to get a disease severity score that was taken accurately. We had a scoring sheet, and the technicians in the field, or the veterinarians who were doing this, were shown how to do the score. So it was very, very uniform. Often if you're just relying on producer records, it's hard to get uniformity there. But if you look here, these were the low responders, and they got the highest score in terms of severity. And so what this means, and we've seen this over and over again, a high immune responder animal, if they get sick, they tend to have, really minimal clinical signs. It doesn't tend to be that severe. And so you may not even need to use antibiotics on those. You maybe could choose something else. But the low immune responders, when they get sick, they get pretty darn sick, and so you do have to use antibiotics. So there's this other tool now. You see, you can begin to take a genetic technology and apply some management information on top of that, because now you know who your females are, and when a low immune responder gets sick, you might as well treat it right away or cull, right? And now here's just the other thing. I want to show you something similar, but this now comes from Immunity Plus daughters. So this, of course, is where we're actually, if I go back, that's where we're actually doing the phenotypes on the animals themselves. And this is information on Immunity Plus daughters. So that means when you're using the Immunity Plus semen. And this now, these health records would have been coming from the producers themselves. And this was from 35 commercial herds totaling 30,000 Immunity Plus daughters. That's the other thing about technologies. You know, sometimes you can have a great technology, but it doesn't actually scale up. And so that's something we were concerned about and wanted to be very sure about. And a slide like this just shows you how well this technology scales, because you can see here's the disease prevalence in Immunity Plus daughters versus non-Immunity Plus, and you can just go all the way down the list here, and you can see overall, if you add all of this up and take the average, you get a 12% less disease in these Immunity Plus females. And this is the paper, by the way, right here by Larmer et al. That's Larmer and Mallard in cattle practice. And so you can get more details about that. But again, you know, mastitis, persistent mastitis, pneumonia, diarrhea, these are areas where we might traditionally use an antibiotic. And if the more high immune responders you have, the less antibiotics you need to use. Here's the picture actually of response to commercial vaccines. I'll just put that in here because I guess if you can get an added benefit in terms of vaccination, and this particular vaccine is J5 E. coli, and it is an anti-mastitis vaccine. And the green line here shows you the response. So this is when they are vaccinated here, week -8. And you can see these high immune responders have much better response, and they keep their antibody response longer than these average or low immune responders. So you're getting that added synergistic benefit. Again, an opportunity to use less antibiotics because your vaccines are working far, far better. So that's another way. I guess that kind of takes us to the end of that section.
Yeah, and it's interesting that, you know, how people, how our customers are using that Elevate test to identify their high immune responding animals, first, second, third lactation cows, and beginning to save that colostrum just to give, you know, the newborn calves on the dairy that little extra head start. So I think it's a great slide to illustrate that. Peter, any quick questions here before we move on to the last section here?
Yeah, there's a question from somebody who asked, are there any indications that producers are able to realize a better milk price using immunity technology? And I think it's quite obvious. So for me, when you think about less treatment, so less treated cows, you actually will have more sellable milk. So I think that's the answer to the question, but maybe somebody has an additional comment here.
Oh, I think
yeah, I think that's a good question, Peter. We've had a lot of discussions with milk processors in different parts of the world, and there's no question that the more that the processors and eventually the retailers and the customers know about their source of their product and the, you know, management environment that, you know, that product was produced, the more comfortable consumers seem to be. And you know, hopefully eventually that can translate into a value proposition that where some of that additional money for the milk will funnel its way back to the farm as well. But there's no question it really has the interest of the processing and the retail community. Other questions, Peter?
Yeah, so this one is interesting. There's a question from Argentina. It's mentioned in the question, so I thought I should mention that as well. So Juan is questioning, so what's the influence of epigenetics when you have on the improvements in animal immunity? And I think that's a very good question, actually. So, Bonnie?
Yeah. So I guess epigenetics is really the forefront of, I think, some of the next improvements that we're going to see, but we're still developing the tools really that we need to accurately measure epigenetics. And just for those that might not be as familiar with epigenetics, epigenetics are changes that are not in the DNA sequence, but on the structure of the DNA, the chemicals that bind to the DNA and how they influence health or any other parameter that you want to look at. So we see that we have epigenetic differences in these animals, but we're really not yet able to say how we would utilize that information effectively. Hopefully in the future, we'll have a better handle on some of that information.
Okay. Yeah, and here's somebody who wants to get a little bit more clarification on the vaccination. So, is it right to assume that HIR will have less animals that do not react to vaccination programs?
HIR will have more animals that respond to the vaccine. I'm not sure if I got that, or are you talking about any, or are you talking about negative responses to the vaccine?
No, so I think that's somebody who wanted to have clarified. So immunity plus or HIR, the better the immune response is genetically, so also they will respond better to those vaccinations.
Exactly. I mean, they respond better to the pathogens. And remember, most vaccines are just components of the pathogen. And so I think it makes intuitive sense that they would also respond better to vaccines. One other little point I'd like to add here that sometimes people don't really understand is that when you give a vaccine, there's always a proportion of individuals that are non-responders. They just don't respond to those components, or they are very low responders. And so the other thing that we've seen is that the proportion of these non-responders is much lower in the high immune response group. And that's another important thing, because you know, if you're a producer, you don't want to spend that money on a vaccine and have 30% of your herd, which can happen, by the way, not respond at all. So that's just like, yeah, flushing that money down the toilet. So that's definitely another added benefit.
Yeah, and here's a couple of people that want to get some more clarification on the genomic test. So here's somebody stating genomic breeding values differ across countries. How would you advise to use a genomic tested female that is not from your population?
I think Brad, you probably answer that better than anyone else.
Yeah, I think in general the genomic test is going to give you, a good idea of within your herd and within your population, kind of high, low, and average. Whether it gets the specific ranking correct, you're never quite sure, I suppose. But we wanted to design the genomic program so that if you did have a group of low responders, you could make decisions with them. And once you've identified low responders, regardless of the population, you may adjust how they're managed. You may decide to keep them or not, or it certainly may influence how you select sires to mate with them for the next generation. So there are, a number of ways people use that information, but I think it does a pretty good job of getting the high, low, and average regardless of the country.
Okay, yeah. I think we should move on to
the
next
so
point.
Bonnie,
There will be a couple of more questions, but yeah.
Right. So Bonnie, I think the next slide is about what's next.
Yeah, so what's next? It's always exciting to talk about what's next because I have a very full and busy lab with lots of projects going to help make this technology even better. So I've got 5 things that I'd like to tell you about today. The first one's what I call specialized genomics. And so that's adding a genomic element which is specific to the innate host defense. So remember that triangle I showed you, and then there was a little triangle within that was innate host defense. So I had a student, PhD student, who was working specifically on that and targeting a cell type called the bovine macrophage. And so we've now found a way to really get some informative SNPs around that, and we filed patents at the end of his PhD, which was just in this past December, and so we are really excited to be able to hopefully add some specialized genomics to what we're already currently doing. Colostrum is a big one. We've been looking at this for quite some time, and so we have a project on colostrum to really see how we might be able to improve that further and get a product which maintains its bioactivity better than some of the current colostrum products that are on the market. Climate change or global warming is a big one that we're working on, and so we're looking to see if there are certain phenotypes, certain immune response phenotypes that also are better able to cope with heat or to be heat tolerant. And when we find those animals, we call these resilient. And so we have a project that's ongoing in that. Angus beef, we've got a project that's funded by Genome Canada, and we're working with American Angus and Canadian Angus and the Semex Alliance to adapt this high immune response technology specifically to Angus, for Angus breeders, and all the way through to the feedlot. And there we've targeted bovine respiratory disease complex because this is a big issue in the beef industry. It's where the most antibiotics is used, and so we're hoping to make a big impact there in terms of improving beef health. And then we have ongoing project in other species. The big one right now is swine, and we're working with 8 of the largest Canadian swine breeders in Canada, again, to help them improve their herd health. Yeah, so those are kind of the 5 big ones. There's other ones for sure in epigenetics and so on, but I just picked these 5 to kind of highlight for you.
That's great, Bonnie. Thanks. I think we're drawing near to the end of the presentation for today, but Peter, are there additional questions?
Yeah, so somebody is asking me, do you have any idea of the impact of Immunity Plus on parasitic diseases like tick-borne diseases in tropical and subtropical countries?
Yeah, we don't have specific research data on the tick-borne diseases, unfortunately, but we are relying on the underlying premise that the immune system, when you get a robust and balanced immune response, that this should be able to handle any kind of disease that we're seeing. We have got some information on diseases that are prevalent in the Middle East, for example, like pink eye and other things like that, but we don't have anything specifically on ticks.
Okay, thank you. And something a little bit related to that. So somebody asked, does the HIR also have an influence on the prevention of heat stress?
Yeah, can I show you? Can I just flip forward here for a minute and just show you a really neat graphic here that I have with me. I'm just gonna put my glasses on. So you know, this is a protein here. It's called heat shock protein 70. And the job of heat shock protein 70 is to protect cells from heat stress. And so we've just finished a project in Holsteins in our research herd, looking to see if we take cells from these animals that we know their immune response phenotype, so you can see them here, high, average, and low in our color code. And we looked at this heat shock protein. What happens? So on the y-axis here, this is the amount of heat shock protein, and down on the bottom, you get the 3 bars. And here, this TN is under thermal neutral conditions. So this is when they're not heat stressed. And you can see, even when they're not heat stressed, high immune responders tend to have more of this heat shock protein sitting around waiting if it's needed. Then here, we begin to heat stress these cells. And you can see here the high immune responders produce significantly more of this protein. And so what the protein does is it helps protect from cellular damage. It helps protect the cell from dying and proteins from being misfolded. And then we give the cells a rest. They get a 24-hour reprieve, and then we come back and we heat shock them a second time. This is the second treatment. And again, these high immune responders have more of this protein, and then the same at 24 hours after the heat stress, they still have more of this protein. So we're quite encouraged about that. And we did have another test going in beef cattle, and I thought this was fascinating and kind of threw it in here in case someone asked about that. And what we did in these, this is mixed breed beef. And what we did is we looked at how they were able to maintain their core body temperature during very hot days when the so-called temperature humidity index was above 74, which is kind of a threshold for heat stress that we use here in Ontario. And so what you can see here, here's the green bars. This is rectal temperature or core body temperature. And this is those animals that are high AIMer, high antibody responders. And this is them under normal THI, normal temperature. So they have a normal body temperature sitting right there, even a little bit below. There's a range that you can have for body temperature. But when the THI is above normal, so during heat waves, for example, these animals do increase their body temperature slightly, but there's no significant difference in that change in body temperature. But if you look here, these are the average immune responders. They do very poorly in terms of heat stress. They get up here with body temperatures are even 39.1, and this is the average, remember. So there were some cows that had body temperatures up over 40. And so this is actually, they're actually spiking a fever and, you having sickness behavior. And here's the low responders. And again, they had increases in body temperature. So to kind of answer that question, yeah, we've got 2 very, very good pieces of information. One in vivo, which you see here in beef, and the other was the in vitro, actually looking at the cells of the immune system themselves and heat shock protein. So we want to do more in this area, but we're really encouraged that you can actually have resilient animals, those that are both healthier and also better adapted to heat stress. So hopefully that answered that question.
Yeah, that's amazing, Bonnie. So, yeah, here's one. Look a little bit at the side effects. So somebody thinks a little bit like, so if a whole herd becomes immunity plus, so like 99% of the cows will be high immune responders, will there be any side effects negatively from this breeding strategy?
Yeah, no, not at this point. So a question that I'm often asked is about things, certain things like allergy and autoimmunity, because this is what happens when the immune system kind of misbehaves or goes awry, so to speak. But first of all, cattle, we don't tend to have allergy and autoimmunity, and so we haven't seen any of those, but that doesn't mean we aren't looking for it. We want to be wise. We want to keep our eyes open to that. But the other thing is the beauty of a selection index, and that's why we keep AIMer and CIMer and innate host defense in balance, because when the immune system is in balance, it's going to work optimally. You're not going to have it firing off with autoimmunity or allergy or those kinds of things. That's what happens if you select just on single trait or if you select just for resistance to a particular disease. And if, for example, let's say as years go by, because it takes a long time, think how many years or decades it took to really, really improve milk production. I mean, we've got lots of room for improvement here before we hit any danger zones at all. But if we saw in the future, just gazing way down the road, antibody was just getting to where that should be optimal, just leave it like that. Well, then you can just put less weight on that trait in the selection index. That's the beauty of having this multi-trait selection index approach that we use. So great question. I definitely don't see any danger and have no evidence of it at all at this point, in spite of the fact of the tens of thousands of animals that we've tested.
Okay, thank you. So I'm not sure how many more questions we will do because we're a little bit over the time yet, but I've got one here that I definitely want to ask. It's a little bit different. So somebody mentioned, you mentioned COVID-19. So many are promoting milk as a good source of boosting human immunity. So do you believe that HIR animals will produce even better milk for humans?
I sure think that's a possibility. The cows themselves are healthier, and I think we know when animals are healthier, the food they produce is better. Now, we are looking at colostrum, for example, and we can see there is even some human application there. So, you know, many people are taking bovine colostrum. Even today, you can get it at health food stores because it has some very special features. And I don't have time to talk to you today about all the special features of bovine colostrum, but for sure, they have some. I can just show you quickly before we finish up here. Here's a slide that just shows you the amount of specific antibody in colostrum in milk of high, average, and low immune responders. This week 0, this is colostrum. And look at this, there's your high immune responders. Look at the amount of specific antibody that's in there. That's when they package bovine colostrum for human use. You want to use high immune response colostrum if you can, because that's the one with all this beautiful specific antibody. And that even stays in the milk. So there's big differences there. So yeah, I could take a lot of time and talk about that, but that's kind of a very brief answer to a super interesting question.
Okay.
Thanks, Bonnie. Good, thank you, Peter. And Bonnie, if you can just switch to that last slide that talks about the next webinar. And while you're doing that, I will thank everybody for their attendance today. We really appreciate you taking time out of your schedule to attend the webinar. We have Semex representatives in every country that'll be more than happy to talk with you about the technology and how it can benefit your management and your herd performance. And Bonnie, to you, thank you for joining us today. It's great to speak with you. I always learn something a little bit new when we go through these presentations together, and I'm sure that the information shared today was very helpful for people to gain a deeper understanding of the technology, the research, and the people behind it, and we really appreciate that. So thanks everyone for joining. Bonnie, Dr. Mallard, thank you. Peter, thank you as well. Have a safe day, everyone.
Yeah.
Bye-bye. Thank you.
Bye.
Yeah, so I think it will be important for everybody to realize that we will have another webinar in 2 weeks from now. Then we will go on a virtual farm tour at Larenwood in Canada. So somebody was asking me a question that we didn't answer yet, but about how do you control, so if genetic improvements brings us to cows producing in time more than 50 liters per day in average, how do I control dry matter intakes and these high production cows? So at Larenwood they're not yet at 50 kilograms per day, but they have high milk production. So I would definitely advise to join our upcoming webinar and go on a virtual farm tour, especially when we cannot visit farms abroad. So
yeah, I'd love to. I definitely will, do that. I don't know, do I need a special invite for that to join it? could Joan or someone send me that link, or do I just go on to the Semex site and I'll find it there?
I think
had an
everybody who
invite.
is attended will get this invite. So yeah,
Oh, okay. That's
Thank
great.
you,
And
everybody.
yeah, that's great. We can talk more about nutrition sometime.